Okay, here we are in chapter 21, our final chapter on nucleic acids and protein synthesis. We've been together through a lot. If you were with me for the first course, which was mostly general chemistry, then we've been together for 2 terms, which is outstanding. This term, of course, has been the organic and biological chemistry portion, and here we are rounding it out with some biological chemistry, nucleic acids and protein synthesis. Nucleic acids are our 4th major macromolecule of biological chemistry. We've already learned about the carbohydrates, the lipids, and the proteins, and now we're ready to dive into nucleic acids. So in this first lecture, our focus will be the components of nucleic acids. I'm guessing you've had some of this before to some degree, hopefully in the past, but we'll try to make it nice and clear this time through if it hasn't been in the past. In particular, we'll try to unravel these nucleosides and nucleotides and what's the difference and all that good stuff throughout this lecture. So please do pay close attention. 1-letter difference makes a huge difference here in nucleic acids. Let's start off with some basic definitions. Nucleic acids themselves are molecules that store information for cellular growth and reproduction. The major types of nucleic acids, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, and both are large molecules consisting of long chains of monomers called nucleotides. Now those nucleotides themselves, the monomers that make up these very large DNA and RNA molecules, consist of a base with nitrogen, a nitrogenous base as they're usually called, a pentose sugar, which is either deoxyribose in the case of DNA or ribose in the case of RNA, and then finally a phosphate group. So there we see a generic example off to the right, some nitrogenous base, the pentose 5-ring sugar, 5-membered ring, 1 being an oxygen, and then the linkage there to a phosphate group. So there you have it, the generic monomer, the nucleotide for either DNA or RNA. Of course, the sugar changes for DNA, it's deoxyribose, RNA, it's ribose, and then the bases will change. There'll be 4 different bases that we can have in DNA, and then one of those changes for the 4 bases that are possible in RNA. Okay, speaking of those bases, here they are. The bases in DNA and RNA, regardless of which nucleic acid we're talking about, they're all derivatives of the heterocyclic amines pyrimidine or purine. If you remember back when we talked about amines and amides, and I mentioned that those 2 would be coming back, well, here they are as promised, and of course, neither one is straight pyrimidine or purine, but rather derivatives. So the pyrimidines are cytosine, which is denoted by a capital C, thymine, which is denoted by a capital T, and again, this is thymine, not thiamine that we talked about in the last chapter as a B vitamin, thymine, T here, which is only in DNA, and then uracil, which is only in RNA. So wherever we have a thymine in DNA, it's replaced by a uracil in RNA. The purines, A for adenine and capital G there for guanine, those are the same in both DNA and RNA. So DNA and RNA share cytosine, adenine, and guanine. In DNA we have thymine, and then in RNA we have uracil to replace the thymine. And just in case you didn't get it, and the few times I've already said it, the DNA bases are cytosine, capital C, guanine, capital G, adenine, capital A, and thymine, denoted by a capital T. So CGA are present in both DNA and RNA. Thymine, capital T, is only in DNA, and in RNA, as we'll see below, it's replaced with uracil. So there we have the RNA bases at the bottom. They look familiar, right? Cytosine C, guanine G, and adenine A. And then, of course, uracil U replaces thymine T. I make a big deal about it because it is a big deal. It's a difference, and really in terms of the bases, it's the only difference between DNA and RNA, so obviously it's going to come up from time to time in homework set questions and exam questions, so it is something that you definitely want to get committed to memory if you haven't already done so. Okay, so the pentose sugars, pentose meaning 5-carbon sugar. In RNA, it's ribose. That's what the R stands for. In DNA, it's deoxyribose, which has no oxygen atom on carbon 2', so that's why it's deoxy, right? It's missing one oxygen. In the early days, I think some of the biologists were calling it dioxyribonucleic acid, and of course that's not correct and not its official name. It's deoxyribonucleic acid because of deoxyribose here. So we have the carbon numbers atom with the primes to distinguish them from the atoms and the bases, right? The base pairs had nitrogens, they were nitrogenous bases, but they also had lots of carbons there as well. So that's why we use the prime system here to denote the numbering of the 5 carbon sugar carbons as opposed to the carbons that are contained in the bases themselves. Okay, so lots of similar names, right? Here we have nucleosides, and a nucleoside has a base linked by a glycosidic bond to C1 prime of a sugar, whether it's ribose for RNA or deoxyribose for DNA. When adenine and ribose form a nucleoside, it's known as adenosine. And there we have a picture of adenosine where you have the ribose there, which is connected to the adenine base, nitrogenous base. So there we have adenosine now. So in addition to that specific reaction down at the bottom that we just mentioned, ribose plus adenine forms adenosine and a water, we have the generic nucleoside formation, sugar, either ribose or deoxyribose, plus base, so one of our 4 bases, again, thymine if we're dealing with DNA, uracil if we're dealing with RNA, forms that nucleoside plus a water because we form it by dehydration synthesis. We connect along that oxygen, connect along that nitrogen by eliminating that oxygen and 2 H's as H2O. Now, I mentioned names are similar here, so you got to pay very close attention, right? Good scientists are people who watch the details. So now we're talking about nucleotides as opposed to nucleosides. So nucleotide has a phosphate group attached to that C5 prime hydroxy groups on the nucleoside. So there you have it. So we had our nucleoside, which was our nitrogenous base plus our 5 carbon sugar, and then on that 5 prime carbon, which had an OH group, it now forms a phosphoester bond with a phosphate group. So that's our big difference there. And so whereas we had adenosine before, now we have adenosine 5 prime monophosphate, AMP as it's known. And there we could also have deoxyadenosine 5 prime monophosphate, DAMP if we were dealing with DNA versus RNA for ribose versus deoxyribose. So here we see the nucleotides of the purines. So depending again, if we have in black the adenosine 5 prime monophosphate AMP, or if you notice along the bottom there of that ring, the OH group that comes off of that ring position, there's in parentheses just a plain H, that's our deoxyribose, right? Replacing that OH with an H goes from ribose to deoxyribose, so therefore in the pink sort of text we have deoxyadenosine 5 prime monophosphate, or DAMP. If we look at our other purine, we have guanosine 5 prime monophosphate from guanine, and then in this case ribose and the phosphate formed by the phosphoester bond. Or if that OH group down at the bottom of the pentose is replaced with a plain old H, we have deoxyguanosine 5 prime monophosphate, or DGMP as it's more commonly known. Here now we've got the nucleotides of the pyrimidines, and so we see cytidine 5 prime monophosphate, CMP, or again if we replace that OH with a plain old H, which is there in parentheses in the pink font, we'd have deoxycytidine 5 prime monophosphate, or DCMP. For uridine, notice we don't have anything below it in pink because, of course, that's from uracil, which only appears in ribonucleic acid, so we only have the ribose form of the sugar in the nucleotide, and therefore it's only named as uridine 5 prime monophosphate, or UMP. And then same sort of deal with our good friend thymine, which is only in DNA, so we only show the deoxyribose sugar, and therefore we would name it as deoxythymidine 5' monophosphate, or DTMP. So there we have it. Those are the nucleotides that we would use to have monomers for DNA synthesis. Okay, so it is a lot to follow. So here's a nice slide here with table 21.1, the components in DNA and RNA, and this is a summary of what they're made up from. So we have our bases, which are A, G, C, and T in DNA, and then A, G, C, and U instead of T for RNA. Our sugar in DNA, of course, is deoxyribose. For RNA, it's ribose. Our nucleosides in DNA are the base plus deoxyribose. Our nucleosides in RNA are the base plus ribose. Our nucleotides in DNA are the base plus deoxyribose plus phosphate, whereas in RNA it's base plus ribose plus phosphate. And finally, a nucleic acid. In a DNA nucleic acid, it's a polymer of those deoxyribose nucleotides. A nucleic acid of RNA would be a polymer of ribose nucleotides. So hopefully you've gotten it all down. It is a lot of similar names and similar structures there between deoxyribose and ribose, but the differences do matter. And the T and U difference, thymine to uracil, is one that we've made a big deal about, I hope. I hope you've caught on to that, and it is a big deal and will come out again and again as we move forward. Now, the good news is there's not so many of these things, right? There's the nucleosides and nucleotides for DNA versus RNA, and that's it. We've covered them all. So hopefully you notice that the name of a nucleoside that contains a purine ends with osine, whereas the name of a nucleoside that contains a pyrimidine ends with idine. So that's how the idine should be easy because that's how pyrimidine itself ends. So hopefully you've caught on there. The names of DNA, we add deoxy to the beginning of their names, so that's nice and easy, just like deoxyribose versus ribose. And the corresponding nucleotides in RNA and DNA are named by adding 5' monophosphate. So there we have our scheme for naming nucleosides and nucleotides. And although we're dealing with very large and complex molecules, we don't have too many names, right? Here we have table 21.2. There's all the names. So if we look at DNA first, the bases are adenine, guanine, cytosine, and thymine. The nucleosides would therefore be deoxyadenosine, deoxyguanosine for our two purines, and then deoxycytidine and deoxythymidine for our two pyrimidines. And of course, once we make the nucleotides, we have deoxyadenosine 5' monophosphate, or DAMP, deoxyguanosine 5' monophosphate, or DGMP, deoxycytidine 5' monophosphate, DCMP, and deoxythymidine 5' monophosphate, DTMP. For RNA, not much different, actually even easier, because we have adenine, guanine, cytosine, and uracil. As long as you can remember the uracil, it's even a shorter name. Adenosine would be what adenine's called, just like up above, only minus the deoxy because we're dealing with regular old ribose now. Guanosine, cytidine, and uridine. And then the names for the nucleotides: adenosine 5' monophosphate, AMP, guanosine 5' monophosphate, GMP, cytidine 5' monophosphate, CMP, and uridine 5' monophosphate, UMP. So it seems a little off-putting maybe at first, but at least this is it for naming. Not like that organic nomenclature we started the course with, where I could put a substituent here, there, anywhere, and you've got to come up with the number and the name and how to deal with that. Here it's what you see is what you get. Sure, they might be pretty long names for the nucleotides, but there's only 4 each in DNA and RNA. Okay, so in particular, you probably thought that adenosine monophosphate sounded familiar, and it should have, because you've probably heard of its offshoots, adenosine diphosphate and adenosine triphosphate, and those are accomplished by adding phosphate groups to the AMP to form first the diphosphate ADP and then the triphosphate ATP, and it's that triphosphate that you probably heard a lot about in your previous biology coursework, because ATP is the energy currency of the cell, right? That's the so-called high energy compound that the cells use as an energy source. So we see it here. It's a triphosphate. Those are phosphoester bonds, right? and we have 3 of those phosphate groups in the adenosine 5' triphosphate, ATP as it's more commonly known. And as we break that bond and form the ADP, well, we can have a net release of energy, right? Sometimes it makes it seem like that bond's unstable and it's explosive even. I see those starbursts in those biotexts, and it bothers me to no end. It's not only true in chemistry, it's true all over the place. Whenever you make a bond, you release energy. Whenever you break a bond, it requires energy. It's just that total sum of energy released by making those new bonds after you take that ATP and bring it back to ADP. That requires energy to break that bond. It's sometimes called the high energy bond. It still requires energy to break it, but the sum of all the new bonding that you make is what gives you a net energy. So just be careful about that. One of my pet peeves, but you know, it's not usually in the chemistry text, but sometimes in the biotext you'll see that sort of makes it look almost like dynamite, these ATP molecules, and that's not true. They are stable, but when they do have that phosphate group removed to form ADP, the new bonds that are formed are what release the energy. Okay, so it's the end of the course. You'll forgive me that little rant right on the last slide. But here we go. We'll end this chapter lecture, as we often do, with a learning check. So for this particular learning check, you're asked to give the name and abbreviation for the following molecule and list its base and its sugar. So hopefully you've been paying careful attention. Go ahead and pause the video while you make those assignments, and then start back up when you're ready to check your work. Good luck. Okay, and hopefully you were able to make pretty short work of this, and you came to the same solution that I have down here. So this is guanine, so therefore because the guanine is bound to ribose, and then that 5' OH group is caused to form an ester, phosphoester linkage there with the phosphate group, we actually have guanosine 5' monophosphate, or GMP. So our base pair there was guanine, and our sugar was ribose, as we can tell by those twin OHs along the bottom, right? That's our quick and dirty method. If we look at that bottom of that 5-membered ring there, the sugar ring, if we see 2 OHs there along the bottom, we know we've got ribose. If we see an OH and an H, then we know we have deoxyribose. So hopefully you were able to get this correct. If you weren't, hopefully it makes more sense now. And if it's just frustrating, unfortunately, this is a lot of memorization. There's not a whole lot I can do, but certainly if you reach out to me for some guidance, I'll do whatever I can to help. Otherwise, hopefully things are all going well and you're ready to move into the second lecture here in our final chapter.